Hello everyone, thank you for joining me today. My name is Jonas Korlach, I'm Chief Scientific Officer at Pacific Biosciences. I would like to thank the organizers of BioConference Live for the opportunity to speak to you today about the application of long sequence reads for more comprehensive views of human genetic variation. My presentation will last approximately 45 minutes, and I will be happy to answer any questions at the end of the presentation. You may submit your questions at any time during my presentation using the Q&A panel you see on your screen. Due to certain limitations with the file conversion into the BioConference Live environment, some of the images in my presentations will appear somewhat blurry. You can access a high-resolution version of the slide deck by either clicking on the Content button on the lower left of your screen, or you can go to our website at www.pacb.com/BCL. And with that, I'd like to begin my presentation. I would like to discuss the value and application of PacBio sequencing in the context of the waves of the different technologies after the initial human genome sequencing. So after the human genome was sequenced for the first time, the first wave was genome-wide association studies, or GWAS, which was focused on finding single-nucleotide polymorphisms, or SNPs, single-base changes in the genome compared to the reference genome to associate them with natural variation and also with disease. And I think it's fair to say that the scientific community has a consensus that that effort had it had some value, but could not explain all the different diseases and all the different associations. So as a consequence, a second wave came to be, which was exome sequencing, now widening the interrogation space of the genome to all the SNPs that are located in protein coding regions. And so with exome sequencing, there was a larger degree of data and larger knowledge. But still, it was found that not all diseases were, it was not possible to associate them with the genetic blueprints. And so right now we're witnessing a third wave, which is looking and finding disease genes genome-wide, identifying the SNPs on a genome-wide basis, whether they're in genes or outside of genes. And so this is the so-called $1,000 genome, and there's no doubt that it will bring about tremendous advances in our understanding of the genetic variation and the associations for disease. But it's also worth noting that there are certain limitations. The $1,000 genome will not include some regions of the genome. They're either not covered or are covered at poor quality. It will not include haplotype phasing, drawing the connection of the SNPs with respect to whether they're contained on the mother or on the father chromosome. It will not contain many forms of larger structural genetic variations that are of a larger size, and it also does not include the myriad of the different gene products that are made from these genes. As an illustration, I would like to cite a few papers that highlight this limitation. This is a paper by a large number of authors looking at the different short-read sequencing technologies and asking about their value for clinical interpretation and implications from these whole genome sequencing events, looking at the SNPs. And the authors concluded that depending on the sequencing platform used, 10 to 19% of inherited disease genes were not covered to accepted standards for single nucleotide variant discovery. Similarly, this is a paper by Hong Li from the Broad Institute looking at artifacts in variant calling from these high-coverage, high-throughput sequencing efforts. Hong Li used a relatively rare haploid human genome cell line sample and asked about the frequency of heterozygous SNPs. So heterozygous SNPs cannot occur in a haploid sample, so they're likely to be artifacts. And he describes in the paper that initially he thought his sample wasn't really haploid because about 10% of the SNPs called by the short-read technologies were called as heterozygous. And so he looked and validated some of these and convinced themselves that they were indeed artifacts, and went further and identified the root cause in erroneous realignment in low complexity regions and the incomplete reference genome with respect to the sample as the two major sources of errors, which press for continued improvements in these two areas. Now, going beyond SNPs, it has been well understood since the first diploid genome sequence of an individual human was deciphered, this was a paper by the group of Craig Venter and colleagues, that non-SNP DNA variation is also very frequent and pervasive through the human genome. So in this paper, the authors described that non-single base, non-SNP DNA variation accounts for about 1/5 of all the events identified in that individual. However, they involve 3/4 of all the variant bases. And this suggests an important role for non-SNP genetic alterations in defining the diploid genome structure. What do we mean by structural genetic variation? These are larger scale, beyond just a single base change in the DNA. They can be deletions where sometimes 10 to hundreds of thousands of bases are missing in the individual relative to the reference genome. Tandem duplication, where a certain region in the genome is duplicated in tandem. Variable number tandem repeats, where these tandem duplications occur at a variable number and frequency. Dispersed duplications, novel insertions, where there is some genome sequence that is just simply not present in the reference sequence. Repeat insertions, inversions, where a piece of DNA is just flipped relative to the reference, or larger scale translocations, where pieces of the chromosome are swapped with one another. The limitations of the short-read high-throughput sequencing devices for discovery of these larger structure variations has been well documented. This is just one example, a paper by Stephen Scherer from Toronto. And the figure on the right shows on the x-axis a log plot of, in this case, insertions. And so on the left you have 1, and that's the SNP, and then going further, a 10-base insertion, 100 bases, 1,000 bases, and so forth. And the colored lines indicate all the published personal genomes that were reported with the different technologies, so 454, Illumina, Solid, and Complete Genomics, comparing them against the black line, which is the human genome reference genome with the Human Genome Project that was done with Sanger. And you can see that in some cases with the 454 and the Illumina technology, the papers on these personal genomes didn't even contain information about insertions above 100 bases, so the curves stop there. For the other technologies, there is some information, but you can see that there is a large gap in the available information relative to the black curve, which we know is true from the human genome sequence. So the authors observed that high-throughput sequencing approaches only detected a fraction of the full size spectrum of insertions, deletions, and copy number variants compared with the previously published Sanger sequenced human genome, and they posit that generating longer reads can mitigate these shortcomings. So in that context, I want to today present to you some of the research from our users that demonstrate that we believe there will be something else after the $1,000 genome, and looking at just the SNPs is not enough. So the fourth wave, we believe, will consist of more comprehensive genome resequencing to comprehensively profile the disease genes with regard to all the different types of variation, not just SNPs, but also structure variation, phasing the haplotypes, looking at the splice variants of the gene products. And then ultimately going to whole genome de novo sequencing, where you free yourself of the reference and possible reference bias to comprehensively profile the entire genome with respect to these forms of genetic variation. So during my talk today, I want to focus on these two areas and show how the long reads provided by PacBio sequencing can make an inroad and can provide high scientific value in these particular areas. I assume that most of you will be very familiar with single molecule real-time or smart DNA sequencing, so very briefly, we observe the activity of single DNA polymerase molecules in real time, and we detect its activity as it's selecting the nucleotides from the solution and incorporating them into a growing strand of DNA. We have the bases tagged with fluorescent colors that we can tell by the virtue of the color which of the 4 bases is being incorporated at any given time. This is done in a highly parallel fashion where we have an array, an ordered array of nanostructures where this reaction is being observed, and in each of those a reaction can potentially happen. This is a view of the full array down here on the lower left, where we have 150,000 of these potential sequencing sites. The activity of the single DNA polymerase molecules is extracted in time to give rise to the raw sequencing trace, and then this is being converted by the software automatically into the FASTA and FASTQ files. All of this happens in the instrument, the PacBio RS2, which is a highly automated sequencing machine. So the first thing I want to mention is that with this kind of sequencing doing it quite a bit differently from the short-read technologies come key sequencing characteristics that also distinguish it from the other sequencing techniques. And the three topics that are important when evaluating really any sequencing technology are listed here. And so in terms of contiguity, that is referring to the read length. You may know that we have by far the longest individual sequence reads. Half the bases that are sequenced, half the data is now contained in sequence reads that are greater than 10,000 bases. Our users routinely see reads greater than 30,000 bases. In terms of accuracy, we also believe we have industry-leading standard, routinely achieving greater than 99.999% or QV50 or for many, many diseases. I've only listed a few of them here on the right. It's responsible for the regulation of the immune system. It's highly polymorphic. Basically, every individual has a different haplotype and the two different alleles. And of course, it's the major cause in organ transplant rejection and also important in disease defense. So this is an area that is of extremely high importance to understand many diseases. The traditional approach to HLA sequencing and HLA typing does not cover the entire HLA gene. This is an example for HLA-A, a so-called class 1 gene that's about 5.5 kb in its overall size. And the traditional typing approaches with Sanger sequencing or with the short-read approaches don't sequence the entire gene. They only sequence exons 2, 3, and sometimes 4. And the phasing, the allelic phasing, making the connection between the SNPs for the different haplotypes is not made directly. It is either done through supplementary methods, for example, site-specific PCR, or when it's not measured directly, it is imputed statistically, that you look in the database and look which of these particular combinations have occurred in the past, and in a nutshell, make that assignment indirectly. And so you're not directly measuring the connection between the different fragments, and it's very difficult to understand whether this A belongs on the same, let's say, maternal chromosome to the C, or whether it's connected to the T. And so precisely this phasing is at the heart of improving the chances of transplant operations and also understanding these diseases. The PacBio solution is full-length, fully phased allele-specific HLA sequencing. Because of the long read lengths that the technology provides, you can sequence the entire gene, all the exons, all the introns, and then we have software that splits out the reads into the two different alleles, calls consensus, and gives a very high accurate full-length, continuous long-read HLA gene sequence for the two alleles. So this is the type of information that you get. The reads and the method requires no statistical imputation. This is an example of a 5.4 kb continuous sequence for class 1 allele that was done from a reference sample from a Japanese user, and it had a 100% match with this particular HLA type. And so that is the reason why several of the HLA typing labs have now gone on to purchase a PacBio system. So, for example, Histogenetics has selected Pacific Biosciences as a platform to do HLA typing, and they're one of the global leaders in this particular field. Similarly, Anthony Nolan, a UK-based blood cancer charity, has purchased two instruments, and they're also going to use it for advanced HLA typing. And with this new capability come about new alleles that have been identified and also correcting the database. So one HLA-B allele has been corrected in the IMGT database, and 4 novel alleles have already been identified, and this was presented by Anthony Nolan, the charity, at the recent big HLA meeting in FE in Stockholm. A second example I would like to highlight is based on taking advantage of both the contiguity and the better uniformity and the lack of the GC content, and this particular example is in the area of Fragile X. The Fragile X gene is at the heart of the most common heritable form of cognitive impairment. That's the Fragile X syndrome that is present at a frequency of about 1 to 3,000, more in males than in females. It's also related to autism and this autism spectrum disorder. It is also associated with other disorders. It's the most common heritable form of premature menopause and responsible for the so-called late onset neurodegenerative syndrome. And so the causative mutation is on the X chromosome in a region that has been extremely difficult to sequence traditionally. And so in a paper entitled Sequencing the Unsequenceable, Paul Hagemann from the School of Medicine at the University of California in Davis describes the ability of single-molecule real-time sequencing to characterize this particular important gene and allele. And in the paper, they describe that smart sequencing provides an approach to sequencing that is fundamentally different from other next-generation sequencing platforms and is well-suited for long repetitive DNA sequences. So this is a figure from the paper showing that with smart sequencing, you can get through these CGG repeats that are expanded as a manifestation of the repeat. So these are the classic trinucleotide repeat expansions where you have sometimes thousands of bases that are 100% GC content, so it goes CGG, CGG, and so forth. And smart sequencing both has the read length to get through this region and has unique anchors associated with the particular individual sequencing reads that allows you to measure the length, and it also has a lack of bias that allows you to get high-quality bases in those contexts. Now, there are non-sequencing based methods that allow you to estimate the length of this particular repeat expansion, but I think Paul Hagemann very powerfully highlighted the necessity to sequence and get the base information for this particular region, and this is highlighted in this particular slide from his talk that he gave at the American Society for Human Genetics meeting in 2012. Because this slide shows that if a mother has 1 or 2 A's interspersed in this sea of CGG repeats, her risk of giving birth to a son that has the full Fragile X mutation goes from about 80% down to about 20%. So 80% with 0 AGGs and 20% with 2 AGGs. So that has profound implications for parental counseling and for prenatal decisions and understanding the outcome of the disease. Trinucleotide repeat expansions are very common in many neurological diseases. These is just a few examples: Fragile X syndrome in a UTR, Huntington's disease, several ataxias, myotonic dystrophy, all can be associated with these particular trinucleotide patterns that are expanded in the diseased individuals. And there are many of these in the human genome. So as an example, trinucleotide repeats, there are about 140,000 instances in the human genome. 32,000 of them have repeats of 6 or more. There are also dinucleotide, tetranucleotide, pentanucleotide repeat regions. Other retrotransposon elements, line, sign elements, and copy number variations, all forms of structure variation that are at the heart of different diseases. I want to give one other example. This is a paper by one of our users in Japan, Shinichi Morishita, looking at short tandem repeats, and he had a paper in bioinformatics characterizing these with both Illumina and short-read technology and with PacBio. And in the paper, he highlights, I think, 2 very important points. One is in this figure, you see in gray the reference genome length of these particular repeats on different types of the chromosome, chromosome 7, 9, 15, and 18. And you can see that in normal individuals, they can be fairly reasonably short, and it might be possible to get at these and sequence these with the short-read technologies. However, in individuals that have the disease, for example, this spinocerebellar ataxia type 31, these regions are expanded to sometimes up to about, this is about 800 bases. And so for precisely the individuals where you want to make a diagnosis, it's getting difficult or impossible to get at this information with the short-read technologies. So for this spinocerebellar ataxia 31, he shows that in the reference genome, you have a sequence of 4 A's and a T that is 14 times over repeated. In the genome in that region for these particular individuals, he doesn't find that at all. He finds about a 3,000 base insertion of some completely different sequence, and he shows in the paper that it's not possible to resolve that with the short-read technologies. He uses PacBio to resolve the canonical structure of these particular this particular region, and he finds that there are these penta and hexanucleotide repeats that occur at variable numbers of frequencies. And the graph on the lower right here shows the length of these indices, J, K, L, and M, the length of these repeat units for 11 different individuals. And the height of the bar indicates the length of these subunits, and you can see that for every single individual, they're different. So these are highly dynamic, structurally polymorphic regions of the genome where you really need the long-read technologies to resolve them and characterize them properly for this particular brain disease. Ali Bashir at Mount Sinai, working with Eric Schott, highlighted that the tandem repeats in the human reference genome are systematically underrepresented. In this graph, you can see that most of these points lie above the diagonal, and so the y-axis is the PacBio data, and the x-axis is the human reference genome. So they sequenced a well-characterized human genome sequence with PacBio, whole genome sequencing, and found that the tandem repeats in this particular sample are systematically much longer than what the, on average, than what the reference would provide. They can make this inference because in the PacBio data, they covered 90% of the tandem repeats spanning 300 bases or more and can resolve this particular structure. Now, why is it necessary to study structural variation? I want to borrow a couple of slides from a talk that Mike Snyder from Stanford gave, because I think they succinctly highlight the necessity to look at structural variation in addition to the SNPs. So he pointed out that it's very common in normal human genomes. It's a major cause for phenotypic variation. It's common in certain diseases, particularly cancer, and it's also now showing up in rare diseases, autism, schizophrenia, and other psychological diseases. Why is it not studied more? Well, it often involves repeated regions. The rearrangements are complex, and they can involve many times highly repetitive elements. And so those are areas where the short-read technologies have more difficulties uniquely characterizing those. This, I think, is a very powerful slide that he is showing where per person there are about 1,000 structure variations that exceed 2,500 bases in size, and they're shown here on this slide with the red hash marks alongside the chromosomes. So it's widespread and is different for the different individuals. This is the size distribution of copy number variations or structure variants in a human genome. And to give you a sense of the scale down here, the event size is in, this is 5,000, 10,000, 15,000 bases. And the short-read technologies can access directly maybe up to 1,000 bases. And with reads that now exceed 10,000 bases and go to 20 and 30,000 bases, with the long-read PacBio technology, there is now an opportunity to access all of these other types of variation. This is a table from a review paper by Evan Eichler and colleagues showing that many behavioral diseases, autism, learning disabilities, schizophrenia, are caused by different types of structure variations, deletions, duplications, and so forth. This is a partial table in the paper. This goes on. The limitations of the short-read technologies to look at these types of variations has been described. This is a paper in Nature Genetics by a large number of authors, including Eric Lander, Chad Nussbaum from the Broad, looking at mutations causing this particular kidney disease. And the authors described that it was not possible to get this with the short-read technologies, but only by cloning, capillary sequencing, and de novo assembly did they find that this particular mutation harbors an apparently independent arising mutation that in sequence that was markedly underrepresented in the massively parallel sequencing data. And so the reason for that was that they were lying in extremely long, about 1.5 to 5 kb, GC-rich, 80% GC-rich, coding variable number tandem repeat sequences in this particular gene, the MUC1 gene. And so the authors conclude that these results provide a cautionary tale about the challenges in identifying the genes responsible for Mendelian, let alone more complex disorders through massively parallel sequencing. At about the same time, this paper was published by Wanda O'Neill and colleagues looking at a gene that's related to the MUC1 gene, that's MUC5AC, important, implicated in cystic fibrosis, in lung cancer, in respiratory diseases. And the paper describes the successful utility of smart sequencing long reads for de novo assembly of large repetitive sequences to fill the gaps in the human genome. So the central exon of MUC5AC had never been sequenced, not even as part of the Human Genome Project. Figure 1 of the paper shows that there is a large, about 50,000 base gap in the human genome reference. And the authors describe the application of smart sequencing to resolve this gap and to characterize the genetic variability between individuals. So this figure from the paper shows on the top the central model that was developed through non-direct sequencing approaches, and then the next 4 rows are an African American individual and 3 Caucasian individuals, and fully resolving this particular region. And you can see 2 things. One, you can see the reason why it was really difficult to study with the short read or even Sanger technologies, because all these boxes and the gray rectangles describe complex tandem repeat regions, and you can see that there is difference between the different individuals in the architecture, the general architecture of this particular gene. Structural breakpoint detection. So deletions are generally regarded to be relatively more straightforward to detect with the short read technologies, because you can look at reads that bridge such a junction. There are several papers that have described the utility of long-read PacBio sequencing for this as well. And I want to highlight one in particular, which is looking at tumor suppressor genes, very important in cancers. And so the authors here at the University of California, San Diego, describe the characterization of certain tumor suppressor genes, such as CDKN2A, for with regards to PacBio sequencing. And so it was interesting to note that the sequence of samples, breast cancer samples, MCF7 is the classic breast cancer sample. It's been studied for literally 20 years. And the structural breakpoint for this very important tumor suppressor, CDKN2A, had not been elucidated, had not been previously characterized in spite of previous efforts, including whole genome short-read sequencing. Also, a non-sequencing method, a SNP array, the estimate for the breakpoint was 15,000 bases away from the true breakpoint that was determined with PacBio sequencing and then validated with Sanger sequencing. And the authors note that the difference may be due to the sign and line repeat elements that mark the region of the upstream MCF7 breakpoints, and they conclude that repetitive sequences are known to confound structure variation analysis and possibly explain why previous genome sequencing studies of MCF7 have not annotated this particular tumor suppressor gene breakpoint. Inversions. Inversions are relatively tricky to get at with the short-read technologies. Again, you're looking at the breakpoints, but Ali Bashir, again from Mount Sinai, I think very powerfully highlighted that it is now much more feasible to detect and characterize these inversions and overcome some of the limitations that exist with the short-read technology. So this is a talk he gave at the American Society for Human Genetics meeting in 2013. The talk's actually recorded, so if you want to listen to the entire presentation, you can go to our blog post. And in the slide, the screenshot that I selected, he shows that inversions can be easily detected with the PacBio technology. And in many cases, he highlights that the inversions are not clean, so it's not like a piece of DNA that's simply inverted relative to the reference, but there are regions where either sequence is deleted or, in this case, novel sequence is inserted at the breakpoints. And so that will make the effort using the short-read technologies futile, because they will not have any reads that are have any breakpoints and junctions. And even the PCR validation, where you design a primer downstream or upstream of this region, will have the wrong size and will not give you the validation. There are other papers that I don't have time to go into where that have been published describing the algorithms and methods to resolve complex tandem repeats with long reads. This is from Mount Sinai. Al-Bashir developing a tool he calls PacMonster, and Evan Eichler in a paper that describes the reconstruction of complex repeats in genomes using the long-read PacBio sequencing technology. So now going to the fifth wave, which we believe will be whole genome de novo sequencing without relying on a reference. I want to give an example of this because we have announced in February the first data set that then gave rise to a de novo assembly. And I want to present our de novo assembly, which is shown here on the far right, in the context of all the previous efforts for de novo assemblies of the human genome over the past 7 years. So starting in 2007 with Craig Venter's genome, that was still done with Sanger, then Illumina and 454, the different technologies, High-seq, MySeq, and so forth. And the graph on the top shows one metric of the quality of these genome assemblies. That's the Contig N50. That describes the contiguity of these genome assemblies. And you can see that over the past 7 years, there really hasn't been much progress. All these genomes are relatively fragmented. The average Contig N50 is about on the order of 100,000 bases or so. And so it took a new technology and the adoption of new algorithms to have a transition to a substantially higher quality and contiguity, about 30 times higher contiguity with a Contig N50 of 4.4 megabases. It's not only the fragmentation of the genome that is a concern, but also the information content that you're missing. This is a paper by Evan Eichler and colleagues looking at limitations of the next generation genome sequence assembly. So he took two of those genomes, the personal genomes that were done with the short-read technologies, and looked and compared them to the Sanger reference from the Human Genome Project, and found that these short-read de novo assemblies were 16.2% shorter, so they missed about 16%. That's about 420 megabase pairs of common repeats. They missed 99% of the validated duplicated sequences, and those were missing from the genome, and that translates to over 2,300 coding exons that were completely missing from these particular assemblies. And they conclude in the paper that high-quality sequencing approaches must be considered in conjunction with the high-throughput sequencing for comparative genomic analysis and studies of genome evolution. Our assembly was recently characterized and reported on in a paper by Adam Filippi and his colleagues entitled Efficient Assembly of Large Genomes Using Single Molecule Sequencing. And in this paper that recently appeared on the bioRxives, he reports reference-grade de novo assemblies of 4 model organisms and the human genome from smart sequencing. He describes that the resulting assemblies include fully resolved chromosome arms and close persistent gaps in these important reference genomes, including heterochromatic and telomeric transition sequences. So in contrast to missing sequence, these sequences provide extra information that go beyond what is known in these model organisms or in the human genome. And so these results demonstrate that smart sequencing alone can produce near-complete eukaryotic genomes at modest cost. This is an example from the figure. This is an example figure from the paper showing the contiguity of the PacBio de novo assembly, which is highlighted on the left half of each chromosome. These are the contigs compared to the short-read assembly of the same sample, which is on the right half of each chromosome. And the red dashes, which are a little bit hard to see, you can see them better in the high-resolution PDF version, are putative gaps that are closed even compared to the latest GRCH38 reference. So this particular assembly has over 10 times better contiguity compared to short read and even the original Sanger assemblies. The contig N50 is 4.3 megabases. On average, there are less than 100 contigs per chromosome. Just as an example, the MHC region, where HLA is situated in, a very important region with the short read assembly, was in 60 pieces. In the de novo PacBio assembly, it's in 2 pieces. And as I mentioned, the assembly potentially resolves immediately 51 annotated gaps in the existing human genome reference. A couple of examples for this type of ability and resolution. These are, again, large tandem repeats. This is a gene called filaggrin, a very important gene where mutations cause dermatological diseases, dry skin, eczema. It's also involved in asthma. This is a very difficult gene to sequence. There's at least one user I know of who purchased the instrument just to study basically this particular gene. It's highly repetitive. It has, in the human genome reference, this gene has 11 1 kb repeat units. And in the sample that was sequenced with PacBio, there are 12 repeat units. And so you can see that there's an extra copy in this particular dot plot representation. And it's worth noting that just a single long PacBio read resolves this particular structure uniquely. On the right here, you see a single 16.3 kb PacBio read. And you can see clearly there are 12 copies of this particular 1 kb repeat unit. Lastly, the KIR region. This is the killer cell Ig-like receptor region. It's a very important region in immunology. And like after HLA, one of the most important ones to understand immune responses and so forth. This is a nightmare region to sequence, even with Sanger. And in the de novo assembly, it's fully resolved right here. And the reason why it's difficult is because it's very large. It can be up to 200 kb in size, and it contains repeat units that are on the size of 12 to 17 kb. So then this is a description of the KIR region on chromosome 19. It's associated with responses to infectious diseases, autoimmune disorders, cancer, pregnancy-related complications. It's highly variable in gene content and allelic polymorphism, and it's full of tandem repeats of these highly homologous genes. And Dane Garrity from the Fred Hutchinson Cancer Institute presented a work that at the American Society of Human Genetics meeting in 2013 entitled Complete Resequencing of Extended Genomic Regions Using Phosmid Targeting and PacBio's Smart Long-Read Sequencing Technology, where he contrasted his previous effort that took about 2 years to characterize this particular genomic region with the PacBio technology, where within 1 week he was able to fully elucidate and assemble this particular region, this very important KIR region. Other efforts in terms of the bioinformatic analysis are ongoing. This is a talk that Gene Myers, so you know Gene Myers from the original Human Genome Project. Gene Myers then left sequencing, but then re-entered and now is using his 2 PacBio RS2 instruments for de novo assembly of human and other genomes. This is a screenshot from a talk he gave about a month ago at an award lecture in Boston, where he describes assembling the human genome with an assembler that he developed in 4 days, wall-to-wall, with a standard computational cluster. No step of the process takes more than 16 gigabytes of memory. And so he got an assembly with a contig N50 of 5.4 megabases, so even higher than what we and Adam Filippi were able to achieve. And so this demonstrates that also the bioinformatics part is now there to make the high-quality de novo assembly of human genomes feasible. In the last 15 minutes of my talk, I want to highlight that, of course, the genome assembly is only one part of getting at the information that's necessary to understand the genomes and understand biology. In addition, it is important to understand all the gene products, both with respect to their frequency and with regard to the diversity of the different isoforms that are created from these particular genes. And the difficulties of resolving the transcript isoforms has been described extensively in the literature. On the left here is a commentary by Ian Korf comparing the determination of the transcriptome to buying a magazine, putting it through a shredder, and having an army of bioinformaticians then spending a lot of time and effort trying to put that back together. Also, Mike Snyder on the right commented in Nature Methods that it's really difficult way to do, to take the transcriptome, shred it into pieces, and then trying to put it all back together. And the underlying reason, of course, is that the transcriptome is extraordinarily diverse and complex. You can have alternative first exons, last exons, alternative splice junctions, cassette exons, where only one or the other, but not both of the exons are ever present in a particular mRNA and so forth. And so down here, this diversity of transcripts is actually not a cartoon. This is just one gene, one of my favorite genes, TPM1, for which all these different isoforms that were displayed schematically have been found. Knowing about the splice isoforms is also important for understanding function. This is a very classic example of BCLX, where you have one gene, two different isoforms have opposite biological effects. So if this particular exon is retained, then this gene product inhibits apoptosis or inhibits cell death. If that particular exon is spliced out, the gene product has the exact opposite function in that it activates cell death. The limitations of the short-read technologies to highlight and resolve transcripts has been studied more systematically. This is a paper by Steiger et al. In Nature Methods, where they looked at 25 different experimental methods and 14 computational algorithms with regard to their ability to interrogate the transcriptome and reconstruct splice variants. And the authors conclude that because of the complexity of higher eukaryotic genomes, there are severe limitations on transcript recall and splice product discrimination that are likely to remain limiting factors for the analysis of current generation RNA-Seq data. Even when all the constituent elements are identified, the assembly of complete isoform structures poses a major challenge, and the authors conclude that ultimately the evolution of RNA-Seq will move toward single-pass determination of intact transcripts, which third-generation instruments will realize. And I want to give you a couple examples also from the research from our users that show that this transition is now coming to pass with the use of long-read PacBio. We have developed protocols that are available on our website, adapting the kits that are available for making cDNA libraries, like the CloneTech Smarter Kit, to take poly-A mRNA and turning that into full-length SMRT templates that now contain all of the coding sequence, all the exons, and the 5' UTR, the 3' UTR, and a part of the poly-A tail. This is then sequenced in the PacBio RS2, and as you know, it goes around the adapter and then results in full-length cDNA reads that we call reads of insert, and those are then subjected to an informatics pipeline that we've also developed that takes these isoforms, builds clusters, and calls consensus from these clusters that then result in the final isoforms that go into the evidence-based gene models. First, a couple of targeted examples. This is a paper by Thomas Südhoff, who in PNAS a few months ago, who of course is last year's Nobel laureate, using the gene family of nurexins and looking at the alternative splice patterns mapped by single molecule long-read mRNA sequencing. So this the nurexin gene family is very important for neuronal development in the brain, forming synaptic and presynaptic connections. And it was very difficult to study because the splicing patterns are very complex. So the authors targeted the particular nurexin genes, did an RT-PCR, subjected it to smart sequencing, and then looked at the isoform patterns. And this is a figure from the paper. Up here you have one particular nurexin protein, that's nurexin 1 alpha, with the overall domain structure. Then in the lower portion here, the particular rows are the different exons, and then the particular columns are the different exons, and the individual rows represent the different splice isoforms. So a couple of things worth a note is that there's exon 17 here, which is annotated in red. This was previously not discovered, so this is a new discovery from the paper. And then, of course, you have each of these rows are different isoforms, And it's possible to then, because it's a single molecule technique, to just simply count the number of transcripts that give you information about the relative abundance of these splice isoforms. So in this particular experiment, almost 250 uniquely alternatively spliced isoforms were found for just this one protein. Another example for a targeted approach is in cancer, looking at gene fusions. This paper in Nature Genetics looks at the fusion of TTYH1 with a particular microRNA cluster, and the authors had hypothesized this particular fusion through the high-throughput short-read technologies, and then used PacBio long reads to obtain full-length cDNA reads that directly confirm on an individual molecule that that particular fusion is present, and it resolves the various isoforms that can be differentiated by the different reads. There are, of course, many gene fusion events that have been characterized and associated with cancers. Currently, there are over 200 curated gene fusion events in cancer. And many cancers are associated with missplicing, alternative splicing, splicing mutations associated with variable phenotypes. And there's a large number of diseases that have an underlying cause in missplicing, or diseases that are associated with changes in the relative levels of alternatively spliced isoforms. With regard to looking at the whole human transcriptome, the first paper that we know about is a study by Mike Snyder in Nature Biotechnology that shows the feasibility of deep sequencing full-length RNA from complex eukaryotic transcriptomes, the human transcriptome, at the single molecule level. I want to highlight a paper that came out a few months later in PNAS, also by a group at Stanford, by Wing Wong, looking at what is arguably the best studied transcriptome, and that's human embryonic stem cells. Literally billions of Illumina reads have been used to characterize this particular sample. The authors applied PacBio sequencing, and of the about 13,000 full-length cDNA reads that they obtained, over 1/3 were novel, and it also included almost 300 novel genes. So in this figure on the right, you see the pie chart of 273 new genes that previously had not been discovered, and then also novel isoforms in existing genes with novel junctions, novel combinations of existing junctions, and so forth. They looked at the expression of the new genes and found that many of these are differentially expressed in embryonic stem cells relative to adult or fetal tissues. And so these genes are likely involved in the perturbance of the pluripotency-associated gene networks. And so the authors conclude that the mere process of gene identification, even in these well-characterized human cell lines and tissues, is likely far from complete. This is an example of a novel multi-exonic RNA in stem cells. Here you have the full-length PacBio reads that, in conjunction with their isoform detection pipeline, gives rise to two very confident isoforms. This particular intron hasn't been spliced out in these two reads. In contrast, the same region shown here with the Illumina splice junction reads in red, and then this gives rise to the cufflinks predictions, and you can see that the region is fairly messy and cannot be interpreted. The authors in the paper also make a final point that I think is very important, in that even though hundreds of RNA-Seq data sets are being generated, their interpretation depends critically on the completeness and reliability of gene and isoform annotations on the species and cell types being analyzed. And so I've made a little cartoon to illustrate this point. So if you have your short-read RNA-Seq data, the assignment to the particular genes and isoforms critically depends on the library, the reference that you have for these particular genes. And so there are two corner cases. Case one is where you're missing certain isoforms from that reference catalog. And so then what will happen is that short reads that come from this particular isoform will be misassigned into isoform 1, and you will overestimate the abundance of those particular isoforms. More commonly, on the flip side, the converse is true, where people pool different tissues and organs just to find out what are all the possible isoforms that are in this particular organism. However, in a particular organ, brain, liver, heart, not all of those isoforms might be expressed. So now you have an ambiguity of the short read data with respect to not being able to assign uniquely where they go and where they come from with respect to the isoforms, so that you will underrepresent the true abundance of these particular gene products. And so coming back to the paper, this is a figure from the paper that shows this, I think, very powerfully. In blue, in the scatter plot on the right, showing the gene expression quantification of the transcripts without the inclusion of the new isoforms that were identified in this paper, compared to orange, where they were included. And you can see that there's a large change with respect to the gene quantification to more accurate RNA-Seq short-read data. And then finally, this is the first paper describing a personal transcriptome, again from Mike Snyder's lab at Stanford a few months ago, where the long reads now facilitate identification of the origin of the particular transcripts. And the figure here shows just one example. So in red, you have all the full-length cDNAs from one allele, from the maternal allele, let's say, and then blue from the paternal allele. And because you have full-length reads, you can take advantage of very small SNP differences between the two different alleles and cluster them with respect to their chromosomal origin. And then very interestingly, you see that patterns emerge that show allele-specific splicing. So in the maternal allele, this particular exon is almost exclusively spliced out. There are only two transcripts that have it retained, whereas in the paternal allele, about half of them still have this exon retained. So giving a much higher level of resolution and insight into gene expression and the complexity of the gene products. So with that, I'd like to summarize. I hope I've given you a good example and overview over how new technological capabilities, the longest sequence read lengths, the highest consensus accuracy, the lack of sequencing bias, and then also a unique capability that I didn't have time to talk about, directly looking at base modifications, can lead to more comprehensive views of genomes, transcriptomes, and epigenomes. I want to briefly mention one other application that I think is very exciting, which is in the area of gene therapy and genome editing. I indicated this in the abstract. This is a paper by Matt Porteous and Eyal Hendel from Stanford University showing that the outcomes of genome editing experiments can be much more cheaply and much more accurately now be quantified with the use of smart sequencing. Gene therapy and genome editing, of course, is a very exciting field of research. There are a lot of people doing it, and it's easy to perform these kinds of experiments. It's much harder to actually figure out what actually happened. And so in the paper, they show that with the longer reads, it's very easy, fast, and inexpensive to quantify the genome editing outcomes. It's broadly applicable to any cell type, adaptable to any target site. It works for all the common nuclease platforms, allows for optimization of the gene targeting parameters, and because of the read lengths, it can detect larger side effects, insertions, deletions of these particular gene editing events. So with that, I will stop, but not before expressing my sincere gratitude to all of our users, the scientists who have invested in the technology and are spending efforts and time utilizing the technology, driving the technology forward. We now have over 100 instruments around the world, and we are deeply indebted and grateful to all the scientists who are applying the technology. And a manifestation of their work is over 200 peer-reviewed publications now that have appeared in the various different application areas, and I encourage you to look those up. They're all available on our website, and hope that you find them interesting and adaptable to your research. And finally, I'd like to thank everyone at Pacific Biosciences. It's been just a tremendous honor to present the technology developments that are then used by the scientific community to you today, and they work extremely hard every day to hopefully make this technology useful for your research as well. So with that, I will stop, and I would like now to open the talk to questions. If you have not already done so, you can submit your questions using the Q&A panel you see on your screen. So the first question, I'm going to read the questions off, and then I'm going to answer them. The first question is regarding HLA, and it says, is HLA typing only applicable to large-size DNA sequencing? What are the platforms available in case of mRNA? So the HLA typing that the PacBio solution that I mentioned is pertaining to taking the full-length gene. You're not obliged to that, so it depends on the primer pairs that you choose, whether you're looking at the whole gene or a part of a gene. The class 2 HLA genes tend to be much larger, and so we have examples where we've done the full length that can be up to 11 kb, but if the researcher is interested in the subsection, then you can also do a 4 or 5 kb. With respect to mRNA, that's something that I talked with members in the scientific community that will be very interesting, and it should be, again, depending on the primers, relatively straightforward to target these HLA transcripts and then sequence them full length to look at potential isoforms in HLA RNA. The second question is how HLA typing overcomes the difficulty of 100% GC content containing sequencing and its accuracy. And as I mentioned, in smart sequencing, we have not measured any appreciable bias with respect to that GC content, and so you can sequence through those regions and get very high quality, just like with the normal sort of more average and intermediate GC content, and that allows you with PacBio to get full-length information. The third question is, has Fragile X been analyzed for peripheral conditions such as multiple sclerosis, Huntington's, Parkinson's, and Alzheimer's? And to my knowledge, those kinds of studies are underway, and I think there are, based on these new capabilities, I know that several researchers are looking at this area. To my knowledge, that hasn't been published yet. I think there may be something at the upcoming American Society of Human Genetics meeting. I've seen some abstracts that may talk about that. That meeting is in October in San Diego. The next question is, can smart sequencing facilitate de novo completion of the germline sequence of the lamprey genome? And I would say that that should be possible with regard to conditioning that you can isolate DNA, high quality, long fragments. I should note that the DNA quality requirements are a little bit higher in smart sequencing because we're not amplifying the DNA. Amplification is the great unifier with regard to DNA quality. However, it comes at a cost of amplification bias, and you drop out certain regions. In smart sequencing, we can sequence amplicons fine, but it's not required. And for these de novo projects, we don't recommend it because we see that if you sequence the native DNA, you get much less bias. The DNA stays in a double-stranded form at all times and is never denatured. And it also potentially then gives you access to the epigenetic information as a product of the smart sequencing. Okay, I see that the time is expired, so then that will be the last question we have time to answer today. If you have additional questions, please feel free to email me directly at jkorlach@pacificbiosciences.com. And thank you very much again for attending, and have a good day.